Alright, so next up we're going to take a look at a dihybrid cross. So I'm going to come back to this Punnett square practice page, and I'm going to look at number 3 and number 4 on here because they're going to be dihybrid crosses. So to start off, let's read the question again. So number 3 says, in pea plants, yellow seed color is dominant over green seed color. So that's probably something I'd underline. That's an important thing to know. And it also says that round seed shape is dominant over wrinkled seed shape. So now I got two different traits here. I've got a seed color, and then I've also got a seed shape. I'm gonna have to keep track of both of these in the same Punnett square now. So we're starting off here. It says if a plant that is homozygous dominant for both traits pollinates a plant that is homozygous recessive for both traits, and then it wants to know a whole bunch of stuff. So to start off, we're going to do just like what we did last time. We're going to start off with the genotypes of the parents. So homozygous dominant for both traits, and let's just set up some letters. Might not be a bad idea to set up a key for something like this. So what I'm going to do over on the board here is I'm just going to set up some letters. I'm going to go with a big Y being used for yellow, and I'm going to say a little y is going to be used for green. For round, make that a capital R, and for wrinkled, I'll make that a little r. So again, the letters that you choose aren't necessarily like super important. You could go with A's and B's if you want, but you want ones that look different, uppercase versus lowercase. So I'm going to choose Y's and R's here. So to start off, I know that I've got homozygous dominant for both traits, and I've got homozygous recessive for both traits. So genotypes of my P1. So P1 genotypes, I've got that as my starting genotype. So homozygous dominant, two big Y's, two big R's. Homozygous recessive, two little Y's, two little R's. My next step in this is figuring out gametes. So to figure out gametes, what I want to do for this time is I want to think about the fact that meiosis, when it makes gametes, it includes a copy of every single gene that an organism has. So I have to include both a Y and an R in every possible gamete or every possible combination. So the good news is here at the beginning that there really is only one combination. There's only big Y's and big R's for parent number 1, and there's only little Y's and little R's for parent number 2. So it doesn't matter how I put them together, it's always going to come out with a big Y and a big R. So gamete-wise, there's only one possibility here. So kind of like what you saw with the monohybrid cross in the first one. So what we want to do here now is we want to create a Punnett square off this. So if I know what my gametes are, I know what my Punnett square has to be, because remember I'm making my Punnett square off of my gametes. So if I've got a big Y and a big R, we'll call that parent number 1, put that along the side. Parent number 2, we're going to put it right across the top. Now when you're doing this, a couple things you want to do. Number 1, you always want to put dominant alleles first if they show up. And number 2, you want to keep like alleles together. So what I mean by that is I want my Y's together and I want my R's together. So I'm going to put this in this box as a big Y, big R, little r. So I have both sets of letters together in one box. That's important here. Don't separate your letters. It'll make things way confusing, and really it doesn't work so well. So in terms of figuring out, what do I got here? I have just one genotype. So my genotype is a big Y, big R, little y. Phenotype-wise, how is this going to look? It's going to be a yellow and round plant. So it's going to have some yellow seeds with round shapes. So in answering some of these questions, so we'll look at B through I here. So in answering B through I, first one asks, what's the chance of them being yellow-seeded? So it's just asking about yellow-seeded, and we said that we have a guarantee that all of our plants here are going to be yellow-seeded, so we'd say 100% for that. For green-seeded, none of them, right? So the gene for seed color was going to be heterozygous here, and so that meant none of them are going to be green. For round, it's going to be 100%, and for wrinkled, it's going to be 0%. For the same reason here, because our genotype for seed shape was heterozygous, so it means they're all going to have that dominant type of a shape to them. For F through I, now we're looking at both, so you have to be both. So now you have to be yellow and round. So what we had found is that in looking at this dihybrid, all of them were going to be yellow and round. So we'd say 100% for F. For G, yellow and wrinkled, well, we did say all of them are going to be yellowed, but none of them are going to be yellow and wrinkled at the same time. So we'd have to say 0% for that. And then for H and I, we're both going to have to say 0% because we never saw anything that was green here in the F1. So like we did before, now what we're going to do is we're going to take and use the F1 generation now as parents. So I'm actually going to show you this work real quick because I'm going to have to erase the board. So take a quick look at it, kind of get a feel for what I did here in terms of starting with the parent's genotype, Punnett square, and then kind of analyze the Punnett square. Those steps are always going to be the same regardless of how you start, regardless of whatever we look at later. Your steps for a Punnett square won't change, and that'll be convenient for you. So next up, we're going to take the F1 generation. They're going to be the parents. So there's my starting genotypes for my parents, just like I always do. My next step is to make gametes. Now here, like I did before, I got to put all my different Y's with all my different R's in combination together. So the way you do this, it looks kind of like foil in math class, but so we kind of like use the, when you draw some like arrows, so I can put a big Y with a big R. That would be one combination here. I could put a big Y also with a little r. That would be a second combination here. I like to put circles around my gametes so I just kind of can separate one from the other. And looking at this, there's no other way of putting big Y with any other R combinations. So now I'm going to move over to little y. Little y can go with a big R, and little y could also go with a little r. So there should be 4 combinations then of gametes that parent number 1 can make. Now the good news here is that parent number 2, because they have the same genotype, makes the exact same gametes. So there's going to be 4 possibilities, and they're the same possibilities for this parent. So now that I know what my gametes look like, what I want to do next, I want to make my Punnett square. But remember, gametes are going to determine how big my Punnett square is. So now I've got 4 gametes for each parent. I need a 4 by 4 Punnett square. So this one's going to be bigger than the last one that we saw. Okay, so there's my Punnett square. I've got all my 16 possibilities in there. I got to evaluate and figure out what each one of these mean. So I'm going to do just like I did before. I need a system though. So I'm going to start up in the upper left-hand corner. I'm going to check them off after I've recorded what the genotype and what the phenotype is. And we're going to see that there's some redundancies here. There's going to be some things that are going to show up several times. So we'll start with genotype. So over here, when looking at the total number of different genotypes, first genotype I see is the homozygous dominant for both. So there's just one of those. I'm going to move over to the next one, and I'm going to see that I've got homozygous dominant for the Ys and heterozygous for the R's. I see that here, but I see the exact same genotype here. So I'm going to put 2 check marks in there, and now I've taken care of both of these boxes. If I come over one spot, now I am heterozygous for the Y's, homozygous dominant for the R's. I see that here, but I also see that here, so I can check both those off as well. Finish off the top row. I've got a big Y, big R, little r, and that one goes right down the diagonal. So 1, 2, 3, 4. 1, 2, 3, 4 check marks. Okay, so far I've taken care of almost half my boxes. So I'm going to come back up to the second row. I've already taken care of the first one, so I'm going to go over to the second one. So I've got 2 big Y's, 2 little R's, tally mark, and now he's taken care of. Okay, if I'm looking, I don't think I see him anywhere else. So I'm going to come over to this next one on the second row, this last one on the second row, big Y, 2 little R's. So I see this one here, and I also see this one here. So that takes care of those. So now my first row is all done, my second row is all done, my third row. So I've got the first 2 boxes that I've already taken care of. It's the third one that's left over. So I got a couple of little Y's, a couple of big R's. I see that once. If I look around, I shouldn't see that anywhere else on my Punnett square. So to finish off my third row, I have 2 little Y's and a big R and a little r. So that is here. It's also here as well. So that's 2 spots for that. So then finally, my last box is 2 little Y's and 2 little R's. And that's the only box that's left, so there's just one of them. Now to check my work here to see if I did this correctly, what I want to do is count up my total tallies, and what I should come up with is 16, because there were 16 boxes. So if my tallies equal 16, I did good. So I have 3, 5, 9, 10, 12, 13, 15, 16. So I got all my tallies in spots, so I'm okay. I've done everything I need to do with my Punnett square. Next thing I need to do is I need to figure out what the genotypes mean in terms of a phenotype. So to do that, I'm going to do it over on this side, but I'm just going to use what I did phenotype-wise or genotype-wise in order to determine phenotypes. So first one I see the genotype is homozygous dominant for both. So that is going to be a yellow and a green, sorry, not yellow and green, but yellow and round. Okay, so that takes care of this one, right? So I just put 1 tally mark there, it took care of that genotype. Next one down is homozygous for yellow and heterozygous for round, but still going to be round. So I'm going to add 2 more. Go 1 more down, it's still yellow, still round. So I'm going to add 2 more tallies in. That took care of this genotype. Next guy down, so the heterozygous for both. So still yellow, still round. So so far, I've got 9 that I've looked at, and all 9 have been yellow and round. Next genotype down is homozygous for yellow, and then homozygous for wrinkled. So this is one of the traits dominant, and the other trait is recessive. So one of those there. Come down 1 more. Heterozygous for yellow, and still homozygous for wrinkled, so a couple more of those. So now I've got 3 that have one of the traits dominant, one of the traits recessive. Next one down, homozygous for being green, and homozygous for being round. So now this is still 1 trait dominant and 1 trait recessive, but it's the other traits. So far, that was 1. Takes care of this genotype. Next one down, I'm homozygous for being green, heterozygous for being round, but that still makes me round, so that's 2 more I can add up to my green and round. And then finally, the last one that's up here, my last genotype was homozygous for being green and homozygous for being wrinkled. And there was just one of those. So, as long as my numbers on my phenotype chart add up as well to 16, I know I did good. So this is 9, this is 3, 3, 1. So what that means then is that 9/16 out of all of my pea plants should come out to be yellow and round. 3/16 should come out to be yellow and wrinkled. 3/16 should come out to be green and round, and 1/16 should come out to be green and wrinkled. So to come back and answer some of these questions. So in letter B, it asks about the percentage of the F1 generation that will be yellow seeded. So when it asks yellow seeded, it doesn't really matter what the other trait is. So 12 out of 16 should come out to be yellow seeded. For green, it's going to be 4 out of 16. So the green and round, green and wrinkled, both those are still going to be green, so that'd be 4 out of 16. For D, for the round seeded, so now what I want to do is come back up and take the 9 and the 3 for both of the ones that are round, so that's going to be 12 out of 16 again. For the wrinkled seeded, that's still going to be 4 out of 16. So for B, C, D, and E, we're just looking at, you had to have one of the traits. You didn't have to have both. Now for F, now you have to be both. So yellow and round seeded. So this is our 9 out of 16. So they had to be both of those at the same time. For G, we had to be yellow and wrinkled at the same time. That's going to be 3 out of 16. H is going to be 3 out of 16 as well, where we want to be green and round seeded. And then finally, for I, green and wrinkled, just 1 out of the 16. So for your dihybrids, guys, that's how they're going to work. You're always going to go through those set of steps where you're starting off by figuring out what the genotype is. Once you know the genotype, figure out what the gametes are. Once you know the gametes, you're going to figure out how big your Punnett square needs to be. They won't always have to be these 16s, but if both parents are heterozygous, then they will be. So once you have your Punnett square, figure out how many different genotypes are present, and then you can figure out what proportions are. Answers here, they can be given in fractions, they can be given in decimals, they can be given, well, I guess percentages even too. So you can take that decimal version, convert it over into a percentage. So any one of those would be fine. So there you go, guys. That's the dihybrid cross.